Hello, everyone, and welcome to today's presentation titled Unlock Cell Therapy Potential with Customizable Cell Isolation and Enhanced T-Cell Activation Solution. This webinar is part of the second annual event in the Cell and Gene Therapy virtual event series. I am Amber Ortega of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by GenScript. For more information about GenScript, go to www.genscript.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing today's presentation, click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby within the event. I now present today's speaker, Vivian Zhang, Senior Product Manager at GenScript. For a complete biography on our speakers, please visit the Presenters tab from the menu on the left of your screen. Vivian, you may now begin your presentation.
Hello, everyone. Thank you for your time today. My name is Vivian Zhang, and I'm the Senior Product Manager at GenScript. It's my pleasure today to introduce to you about our capabilities and solutions for cell isolation and activation to advance your cell therapy discovery and development. So quickly for the contents today, we're going to have an overview of the cell therapy workflow and the technology considerations. And then I'll follow up with the technology platform we have developed at GenScript for cell isolation and T-cell activation using magnetic beads technologies. And then we'll dive deeper into our newly launched customizable cell isolation service and how to use that incompatible with our robotic automation capabilities with our collaborator Multiply Lab. And then in the end, we'll look into our T-cell activation, Reagent, Enceed, and see how it can further enhance your T-cell activation and your T-cell-based cell therapy development. Okay, so as you all know, cell therapies has emerged as one of the most promising approaches in the modern medicine. It offers novel solutions for a wide range of diseases, from cancer to autoimmune disorders and regenerative medicines. The therapies can harness the power of the body's immune and regenerative cells with various types of cells, including T cells, NK cells, macrophage, and stem cells. So it plays a very important role now in targeted treatments. In addition to that, advancement in genetic engineering and in vitro expansion technologies has also enhanced the efficacy of the cell therapies. And some of this have moved forward to get approval to be used in treatment, and more than hundreds are currently in clinical trials. As you can see, the production workflow for the cell therapy is quite long and a complex process using many different types of raw materials and instruments, bioreactors. At GenScript, we're honored to provide an end-to-end solution with our ready-to-use products to support the whole process of cell therapy, including endonuclease for the starting material thawing, our featured cell isolation and T-cell activation platform, our gene editing platforms, including CRISPR, viral vectors, our cell expansion cytokine growth factors, as well as a bunch of analysis and QC products as listed here. If you're interested, please feel free to scan the QR code and learn more on our website. For today, we'll focus mainly on the cell isolation and activation capabilities. As you all know, there are many different types modalities in cell therapy, including T cells, NK cells, stem cells, which means there are different cell targets for enrichment. So choosing your proper cell targets and enrichment strategy is the important part for the cell therapy development. And in addition to that, stringent QC requirements always comes for cell therapies. So it is also important to have that in the mind when choosing your raw material and platform for cell isolation and activation as well. And in addition to that, currently in vivo cell therapy has also emerged, but even with that, the in vitro cell therapy manufacturing is still dominating the market as of now, especially for the multi-gene edited modalities like for the solid tumor treatments. So for choosing your best cell isolation and activation platforms, there are many different considerations, but a few most important ones we try to summarize here. And that includes that it need to deliver high efficiency, which is very obvious. You want the highest purity of your target cell to start with, and the minimum of the other cell types that you don't need. It will be best that it is non-toxic, does not cause any immunogenicity, and best if it is biodegradable. And this biodegradability feature can further help your downstream process and make the product easy to use and streamline your process well, and also can help to eliminate any impurity risk. In addition to that, the cGMP readiness, IND application track record would also be important considerations if you want to further move your cell therapy candidates to the clinical trials. So there are a few different options on the markets for cell therapy, for cell isolation and activation. Many of them are using magnetic beads technologies with different size of the beads ranging from nanometers to micrometers. And there are also protein complex type of product that can do the activation as well. And there are pros and cons of different type of technologies, which you will want to fully evaluate all of them to make your proper decisions. So it will be best that the product can meet all of the key considerations we just talked about, but sometimes not all the products can meet all of them. For example, if the beads are too large, then it definitely need to be removed from the cells. So that will require additional beads removal step from the cell surface, as well as additional downstream analysis and QC process to make sure that there is no residual beads left on the cell surface. And when it comes to if it is a totally soluble protein complex, those type of products are typically very easy to use because it's considered as fully soluble, but the performance and the efficiency of activation or isolation will need to be further evaluated and compared so that you can make the best decision for your workflow. And in the case of the Cytosinct cell isolation nanobeads developed by GenScript, it is the magnetic beads product that is composed of safety materials inside and a magnetic core, as well as a stably conjugated antibodies on the outside. And because of the tiny size of the magnetic beads, it isolates the cells with a column-based technology, and the size and iron content of the beads is fully safe to be used for either in vivo, downstream in vivo animal studies or future infusion into the patient with the GMP grade products. So a quick introduction about the history and capability of GenScript for the cell isolation activation type of magnetic beads product. Actually, before this product offerings, GenScript already have magnetic based product for bioprocessing for many years, including for plasmid purification and protein purification. Currently, we already have more than 15 years of experience in developing magnetic beads products for bioprocessing. And we also have our own built-in GMP manufacturing facility that is compliant with global standard. And it is fully ISO 13485 compliant as well for all the CGMP grade raw materials. At the same time, we also collaborate with our sister company, ProBio, to develop the antibodies. As you know, for the cell isolation and activation, besides the magnetic core, a very important component is actually the antibodies. And the high performance antibodies will be able to deliver the best purity, the best recovery, and the best safety. So with our collaborations, we're able to get the powerful antibody discovery capabilities, multiple clone selection, stability optimization with different conditions, formulation screening, as well as to bring down the high cost for GMP grade. And then with the beads as well as the antibodies, then we are utilizing our proprietary technologies for beads conjugation. Then we follow up with the process development. And then all the products are validated inside as well as with our industry partnerships as well to make sure that it can deliver the requirement for the cell therapy development. So throughout the years, the Cytosinct and Enceed cell isolation activation products have actually achieved quite some milestones supporting our customers globally in their cell therapy development. The products is fully compliant with global regulations and quality standards, including FDA and EMA. It is registered for ISO 13485 for the CGMP grade and USB 1043 as well. It also received and is activated for DMA with FDA by successfully supporting clinical trials, IND applications in US. And recently, we also got clearance for the RX360 audit system so that the products and the facilities are ready for remote auditing in addition to the on-site auditing as well. With all of that, the products can deliver the quality and the regularity that is ready to support clinical trials and IND applications in US and global. And a brief summary of our audit history. We have completed so far 55 successful client GMP audits in our GMP facilities, and there's no critical observations, including customers from global-wide, from US, EU, and China. And in addition to that, we also provide a very strong global supply chain as well to enable the fast turnaround time and delivery of our products with warehouse and the shipping centers across all the continents. Our warehouse and headquarters in US is located in New Jersey. So that can enable to make sure that we deliver the products within 2 business days after ordering online. With that, the products has been trusted and used with our global partner companies for their clinical trials and IND applications. We have successfully supported more than 10 IND applications, including the ones in US with FDA. And we have more than 20 IND or IT applications, and more than 100 IND enabling programs ongoing right now. And last but not the least, key considerations for the cell therapy development and raw material choosing is the cost. From a research recently published in Cytotherapy, more than 50% of the cost come from the reagents and the consumables, and that significantly put a financial stress on the product development as well as the providing the drug and treatment to patients globally. With that, with GenScript capabilities and experience and the strong supply chain globally, we're able to provide the products at very competitive price and to help to significantly decrease the cost of the goods, with more than 1 million savings starting from phase 1, and this can significantly support the sustainability of the cell therapy development and manufacturing, and in the end, enhance the accessibility of this type of therapies to patients. And with that, we are able to offer comprehensive product solutions, including our cell isolation nanobeads with many different type of popular targets for T cells, NK cells, stem cells, and more. And our recently launched customization service, which I'll dive deeper in the next slide, as well as our Enceed T cell activation reagents, and our automated instrument in collaboration with our partner Multiply Lab to enable the robotic automation for the manufacturing, as well as GMP grade or RUO grade columns and tubing sets. So from now, I'd like to dive a little bit deeper into the customization beads service that we recently launched. As many of you may already know, there are more than 400 cluster of differentiation, the CD markers on cell surface in human, and there are many type of cells that could be potentially used as a target for cell therapy development or simply just research, much more than the traditional T cells, NK cells, or stem cells. So to further support that, the research need to start from the purified cells for the downstream either analysis or further expansion. However, it is currently not fully enabled in the market to isolate any type of cells by using magnetic beads in a single step. The traditional method is to use a biotinylated conjugated antibody together with the streptavidin beads to isolate the cells. However, this method will need to use 2 incubation steps. You first incubate the cells with the streptavidin beads and then incubate the mix with the antibodies. This 2-step incubation causes additional time and cell loss in the process. So it is definitely not ideal, especially if you deal with a rare cell population. Any loss in the cell number could somehow impact your downstream application or your research. So with that, we're proudly to launch this new service that we can provide this customization service to conjugate or develop any nanobeads for any cell surface target of your choice. And this is enabled with our strong antibody and protein development capabilities and our strong magnetic beads conjugation capabilities and know-how. With this customized nanobeads with the antibody of your choice, you will be able to isolate or enrich any cell target with one incubation step, which can speed up your workflow and avoid any hassle in your research for the enrichment step. And in addition to that, we are also able to provide this for either RUO grade or GMP grade. So this will enable you to further move your project from the research grade to the next step, manufacturing as well. That's something in the plan. So here is a quick timeline for the customization service. Depending on what is the starting material, the timeline would be different. But the fastest is that we can conjugate the beads within 2 months. If you start from the RUO grade, it typically take about up to 4 months to develop the RUO antibody and the nanobeads conjugation as well. And then if you would also need GMP grade nanobeads development, then we can further upgrade this to the GMP antibody development and the GMP antibody PD tech transfer, and then the GMP nanobeads production process as well, followed up by the stability and safety validation, which then in total takes some time, about 16, 17 months for the whole project. So with this, we're able to provide to you the complete one-stop solution, no matter if you want to start with the RUO or directly into the GMP, or if you want to start from a ready-to-use antibody or just to provide us the sequence. And the beads that we provide will go through the strain quality and the clinical safety assurance before they are delivered to you. And they will be compliant with the quality standard in our CGMP facility to make sure that it is compliant to support from IND to BLA. And in addition to that, I would like to very quickly touch on our collaboration with Multiply Lab to robotically automate our cell isolation platform using our nanobeads as well as the Cytosinct 1000 instrument. I'll just quickly touch base on here, and our collaborator Multiply Lab, Fred, CEO, will give us a webinar shortly after mine, so please stay tuned on that. With our collaboration, we're able to robotically automate the cell isolation step. And with the data, as you can see on the bottom in the Stanford and the Multiply Lab joint study, we're able to showcase that the instrument demonstrated comparable T-cell purity and viability across the fully integrated robotic and the semi-automated process. And so the nanobeads that provided by GenScript as well as the customizable nanobeads would be also fully compatible on the instrument as well as the robotic system as well. Okay, so in the next few minutes, I would like to further introduce the T-cell activation reagent from GenScript as well, which is part of our comprehensive offerings, and that can complement your T-cell based cell therapy development. So here is just a quick background about the T-cell activation signaling and how it happens naturally. As you may know, there are three signalings for T-cell activation. The first one is the engagement of the TCR, T-cell receptor on the surface of T cells with the MHC class II molecule. On the APC, the antigen presenting cells, when an antigen is recognized and presented by the MHC class II molecule. And after that, there's the second signaling engagement required from the CD28 molecule from the T cell engaging with the CD80, CD86 complex from the APC cells. And with the two signaling activation, it will induce the cytokine production and the release from the APC, which will further activate the T cells and thus complete the T cell activation in nature. So what a T cell activation reagent do actually is to mimic this natural signaling pathway by providing the CD3 and CD28 molecules on the T cell on the APC surface. So the Enceed reagent, as shown here, is a nanometer sized reagent with a magnetic core inside, but it does not require any magnet for the processing. Thus, the magnetic core helps to enhance the activation result, but is fully safe and biodegradable. With the CD3 and CD28 conjugation, antibody conjugation on the surface of the reagent, it can engage with the TCR as well as the CD28 from the T cell to complete the first and the second signal in the natural T cell activation. And then in addition, in combination with the cytokines provided in the culture, all the three together will complete the T cell activation. So here are some features provided by the Enceed T cell activation. As just mentioned, it is a fully biodegradable reagent and is compatible with any bioreactor. No magnet is needed and the actual residual reagent does not need to be removed by a magnet at all. The actual reagent can be easily removed by a simple media change. So it is very easy to use in a very simplified workflow. And in addition to that, with the advanced capability of antibody screening and selection from GenScript, we're able to select the best clone to enable the higher percentage of the TSCM and TCM, which can deliver the enhanced memory phenotype, which can potentially enable advanced clinical efficacy. And this is something that is quite important for the T-cell-based therapy development. And in addition to that, we have also seen higher fold expansion using in T cells activated by this reagent. And it is also fully compliant with FDA and EMA and successfully supported IND. So here are some data showing the performance of Enceed activation reagent. The T cells were isolated by the Cytosinct system and then treated with the Enceed reagent and transduced with lentivirus for the CAR production. As you can see, the transduction efficiency indicated by the CAR-positive expression is higher in T cells activated by Enceed than another method, and this was seen in various time points on day 3, day 6, as well as the day 11. Besides, higher fold expansion was also observed in T cells activated by Enceed than another method, especially starting from day 11, which is significant improvement in the cell count number, and this can be quite beneficial for T cells that need to go through a longer expansion time or the program that need a very high cell number in the final product. And last but not least, we also observed enhanced memory phenotype indicated by the subpopulation of TCM and TSCM, and also higher killing efficacy against the target cell line. Here used dotted luciferase cell line was also observed in vitro as well. And it will be very interesting to see if this can further translate to clinical efficacy. And besides all the good performance data, the product is also easy for using to seamlessly scale up for manufacturing. We have enabled the consistent performance in products from RUO to GMP. And here is a data testing in fold expansion to show the consistent performance across different grade of products. And this is quite important when you have the program to move forward to manufacturing in the future, that you will want the performance consistent from the RUO to GMP. And on the right panel, it shows that after a simple wash, there's no residual, actual residual reagent on the cell surface. And that the reagent very safe to use with no additional removal needed. So with that, I would like to conclude my presentation today, and thank you all for your time to listen, and I will welcome any questions.
Thank you, Vivian, for your informative presentation. We will now begin the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just submit a question in the Ask a Question box located at the bottom of your screen. We'll answer as many of your questions as we have time for. Now let's get started. Our first question is there limitation on the customization service of cell isolation nanobeads?
Okay, thanks for the question. Currently, there's no limitations that we see for our customization service of the nanobeads. Basically, the service can cover the customization no matter if you want to start from a provided antibody or protein, or if you provide us the sequence of that target protein or antibody. Either way works, and we can provide the different options of the grades as well.
Great, thank you. Our next and last question is, are Cytosinct nanobeads and Enceed T cells activation regions compatible with other platforms?
Okay, thank you for this question. So the Enceed T cell activate reagent, as mentioned, is a fully biodegradable reagent, so it's completely compatible with any bioreactor. It does not need any removal step. So we don't see any issue using it with any bioreactor. For the Cytosinct nanobeads product, it is a magnetic beads that, because they need to go through a positive and negative selection process, it need to use a column for the capturing of the beads and the cells. So based on our current study and the internal validation, it can be used on either GenScript Cytosinct instruments, or it can also be used on other column-based platforms or instruments from other providers as well.
Amazing, thank you. So that is the last question that we have today. So I'd just like to thank you again, Vivian, for your time and your important research. Before we go, I'd also like to thank the audience for joining us today and to thank our sponsor, GenScript, for sponsoring today's webinar. Please visit GenScript's booth in the exhibit hall after the webinar for more information and resources. Questions submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 1 year until October 15th, 2026. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.